Hello, my amazing tribe. I'm excited to connect with you through this video. Welcome, curious minds, to our video that explores the ins and outs of cyclostomy. Cyclostomy, often referred to as Cyclostomata, is a group of vertebrates that comprises the living jawless fishes, the lampreys and hagfishes. Both groups have jawless mouths with horny epidermal structures that function as teeth called ceratodontes, and branchial arches that are internally positioned instead of external as in the related jawed fishes. The name cyclostomy means round mouths. It was named by Joan Crockford-Beattie. Welcome to the next segment, where we explore possible external relationships and its significance in our journey. This taxon is often included in the paraphyletic superclass Agnatha, which also includes several groups of extinct armored fishes called ostrotums. Most fossil agnathans, such as galespids, thelodonts, and osteostracans, are more closely related to vertebrates with jaws called gnathostomes than to cyclostomes. Biologists historically disagreed on whether cyclostomes are a clade. The vertebrate hypothesis held that lampreys are more closely related to gnathostomes than they are to the hagfish. The cyclostome hypothesis, on the other hand, holds that lampreys and hagfishes are more closely related, making cyclostomy monophyletic. Most studies based on anatomy have supported the vertebrate hypothesis, while most molecular phylogenies have supported the cyclostome hypothesis. There are exceptions in both cases, however. Similarities in the cartilage and muscles of the tongue apparatus also provide evidence of sister group relationship between lampreys and hagfishes. And at least one molecular phylogeny has supported the vertebrate hypothesis. The embryonic development of hagfishes was once held to be drastically different from that of lampreys and gnathostomes, but recent evidence suggests that it is more similar than previously thought, which may remove an obstacle to the cyclostome hypothesis. Several groups of Paleozoic jawless fish have been suggested to be more closely related to cyclostomes than to jawed fish, including conodonts and anaspids. The presence of mineralist elements in these jawless fish, like the oral conodont elements and the armored body covering of anaspids and scutes on other species like Laucinius, suggests that mineralist tissues were present in the last common ancestor of all vertebrates, but were secondarily lost in hagfish and lampreys. Now, let's delve into the intricacies of internal differences and similarities and explore its various aspects. Both hagfishes and lampreys have a single gonad, but for different reasons. In hagfishes, the left gonad degenerates during their ontogeny and only the right gonad develops, whereas in lampreys the left and right gonads fuse into one. There are no gonoducts present. Hagfishes have direct development, but lamprey go through a larval stage followed by metamorphosis into a juvenile form or adult form in the non-parasitic species. Lamprey larvae live in freshwater and are called ammocotis, and are the only vertebrates with an endostyle, an organ used for filter feeding that is otherwise found only in tunicates and lanceolets. During metamorphosis, the lamprey endostyle develops into the thyroid gland. The cyclostomy evolved oxygen transport hemoglobins independently from the jawed vertebrates. Hagfishes and lampreys lack a thymus, spleen, myelin, and sympathetic chain ganglia. Neither species has internal eye muscles, and hagfishes also lack external eye muscles. Both groups have only a single olfactory organ with a single nostril. The nasal duct ends blindly in a pouch in lampreys but opens into the pharynx in hagfishes. The branchial basket reduced in hagfishes is attached to the cranium. The common ancestor of both cyclostomes and gnathostomes went through a genome duplication before their split, and while a second genome duplicatio occurred in the stem gnathostomes, the stem cyclostomes experienced an independent genome triplication. The mouth apparatus in hagfishes and adult lampreys has some similarities, but differ from one another. Lampreys have tooth plates on the top of a tongue-like piston cartilage, and the hagfish have a fixed cartilaginous plate on the floor of its mouth with grooves that allows tooth plates to slide backwards and forwards over it like a conveyor belt, and are reverted as they move over the edge of the plate. Hagfishes also have a keratinose palatine tooth hanging from the roof of the mouth. Unlike-jawed vertebrates, which have 3 semicircular canals in each inner ear, lampreys have only 2 and hagfishes just 1. The semicircular canal of hagfishes contains both stereocilia and a second class of hair cells, apparently a derived trait, whereas lampreys and other vertebrates have stereocilia only. Because the inner ear of hagfishes has 2 forms of sensory ampulla, their single semicircular canal is assumed to be a result of 2 semicircular canals that have merged into just 1. The hagfish blood is isotomic with seawater, while lampreys appears to use the same gill-based mechanisms of osmoregulation as marine telosts. Yet the same mechanisms are apparent in the mitochondria-rich cells in the gill epithelia of hagfishes, but never develops the ability to regulate the blood's salinity, even if they are capable of regulating the ionic concentration of Ca and Mg ions. It has been suggested that the hagfish ancestors evolved from an anadromous or freshwater species that has since adapted to saltwater over a very long time, resulting in high electrolyte levels in its blood. The lamprey intestine has a typhlosole that increases the inner surface like the spiral valve does in some jawed vertebrates. The spiral valve in the latter develops by twisting the whole gut, while the lamprey typhlosole is confined to the mucous membrane of the intestines. The mucous membranes of hagfishes have a primitive typhlosole in the form of permanent zigzag ridges. This trait could be a primitive one, since it is also found in some sea squirts such as Ceona. The intestinal epithelia of lampreys also have ciliate cells, which have not been detected in hagfishes. Because ciliated intestines are also found in chondristi, lungfishes, and the early stages of some telosts, it is considered a primitive condition that has been lost in hagfishes. Without wasting any more time, let's jump into the fascinating world of phylogeny. Arthur Mayashta et al. 2019. Share this video with your friends and spread the knowledge.